And now, let's look at what is the mode of actions of hormones and how do hormones trigger cell signaling. From the physiological points of view, hormones are a carrier of information with the purpose to modulate the functions of certain tissue according to the needs of the body. These tissues are receptors or the target tissues. In general, hormone will be produced by endocrine glands, and the glands will secrete hormones into extracellular fluids, often the blood. The blood will transfer hormones to the whole body, and it will eventually reach the target tissues and bind to the specific receptors. The target tissue will respond and result in feedback mechanism. This feedback will reach the glands to either stop or increase the hormones productions. If hormones productions is increased, it is called positive feedback. However, when hormone productions is decreased or stopped, it is called negative feedback. Now, let's look at types or class of hormones. Do you know how many classes of hormones are there? Based on what characteristic and molecular mechanism a hormone is classified into its class or group? Well, hormones can be loosely classified into classes which include protein-based hormones and lipid-based hormones. The protein-based hormones include the polypeptides, glycoproteins, and amines. Polypeptides are small proteins such as insulin and antidiuretic hormones. Glycoproteins are larger proteins with carbohydrate attached to it. Follicle-stimulating hormones and luteinizing hormones are examples of glycoproteins. Amines are modified amino acids which includes epinephrines and melatonins. For lipid-based hormones, it includes steroid and icosanoid. Steroids are modified cholesterol, such as sex hormones and aldosterone, whereas the example of icosanoid is prostaglandins. What are the differences between lipid-based and protein-based hormones? Both lipid-based and protein-based hormones possess different characteristics and mechanism. Lipid-based hormones diffuses across the membrane and enters into the cell. And inside the cell, the lipid-based hormones will bind specifically to receptor proteins in cytoplasm or nucleus. This hormone receptor complex will then enter the nucleus and bind to DNA and activate gene transcription. This will result in protein synthesis, and eventually protein will be produced. On the other hand, protein-based hormones are hydrophilic, and hence they are not lipid soluble. These hormones will trigger secondary messenger pathway by transmitting signal across the membrane. This usually activates a series of secondary messenger with multi-step cascades. The protein-based hormones may also activate cellular responses by uptaking or secreting of molecules or enzymatic reaction. Now, let us take a closer look of the molecular mechanism of protein-based hormones. Binding of hormones to receptor initiates a series of events which leads to generations of so-called secondary messengers within the cell. The hormone is the first messenger. The secondary messengers then trigger a series of molecular interactions that alter the physiologic state of the cell. These processes is also known as signal transduction. Cell surface receptors are integral membrane proteins, and this protein possesses regions that contribute to three basic domains, which include extracellular domain, transmembrane domains, and cytoplasmic or intracellular domains. Certain types of receptors may cluster together in the membrane after binding to hormone. The interactions of hormone-bound receptor with other membrane or cytoplasmic proteins is the major factor to the generations of secondary messenger and transductions of the hormone signal. So, what is the secondary messenger system in this process? Now, put it in a simple way. Let's consider what would happen if, late at night, Adam noticed a serious car accident and the car was on fire. Adam probably will dial 999 or call the fire department to inform the dispatcher of the incident. The dispatcher would in turn immediately contact and activate a number of firemen and the policemen. The firefighters would then rapidly go to the work pouring off the water on the fire, and the policemen will look for any survivor. Once the fire is off and the victim has been found, the firemen and the policemen will go back to the stations and rest. The responses to the car incident or accidents in some way are analogous to a secondary messenger system involved in the hormone's action. In the scenario mentioned just now, Adam is the first messenger, the dispatcher is receptor, and the firemen and policemen are secondary messengers. Currently, 4 secondary messenger systems have been identified in cells, which include cyclic AMP, protein kinase activities, calcium or phosphoinositides, and cyclic GMP. In all cases, small signals such as ligands that is generated by hormone which binds to its receptor will be amplified within the cell into a cascade of actions. This action will later alter the cell physiological state. Now, let us have a closer look on cyclic AMP secondary messenger system. Cyclic adenosine monophosphate or CAMP is a nucleotide that generated from its precursor ATP under the catalytic influence of the specific enzyme, the adenylate cyclase. The increase or decrease of intracellular concentrations of CAMP is caused by a variety of hormones. The fluctuations affect many types of cellular processes. The activations of a CAMP-dependent protein kinase, which is also known as protein kinase A, is a prominent effect of elevated concentrations of the cyclic adenosine monophosphate. Protein kinase A is catalytically inactive state and it only becomes active when it is binds to cyclic adenosine monophosphate or CAMP. Activations of protein kinase A will phosphorylate some other proteins. And these proteins could be enzymes that are either activated or suppressed by being phosphorylated. The changes in enzymatic activity within the cell will alter its state. So to enhance better understanding of this process, let us put this information together to understand the mechanism of actions of glucagons. In the first step, glucagons will bind to its receptor in the plasma membranes of the target cells. This cell could be the hepatocytes. Secondly, the bound receptor will interact with and through a set of G proteins, and these will activate adenylate cyclase, which is an integral membrane protein. Then, the activated adenylate cyclase will start to convert ATP to cyclic AMP, and this will cause an elevated intracellular concentrations of CAMP. After that, the high level of CAMP in the cytosol will activate the protein kinase A. The activated proteins kinase A will move around the cell and phosphorylate other enzymes. These result in their conformation changes and modulations of their catalytic activity. Now, the cell has been changed. And lastly, destructions of the cAMP phosphodiesterase and the inactivations of adenylate cyclase result in the decrease of the cAMP level. In the above example, the secondary messenger system is clearly explained. The example also shows that the hormone's action was to modify the activity of pre-existing components in the cell.